Okay.
Okay.
Said it first word. Whatever. Next unit, guys, we all have our notes, correct? Now, as you all well know, the note packet on front, this is not where we take notes, so everybody flip to the next page where all the note-taking occurs. These objectives are kind of not in the same order. We're going to talk why cells must be tiny. We're going to talk specifically why. And we're going to understand why cells need to be small in terms of surface area to volume ratio. Notice I forgot the B in there, so I had to add that in after the fact because it was a word salad. So, why must cells be tiny? Let's figure it out. Now there is a little bit of writing at the beginning. I apologize, but I don't think your hands will fall off from the writing. Okay? So, reasons, the nucleus and the DNA. The DNA in the nucleus controls the cell's activity. I will articulate as to why this is why cells have to be small after we write this down. We are running a little bit behind schedule, but that's okay. This controls the activity. Think about it, guys. If you have something inside of a cell, you have this DNA, a certain amount, and you need to make the same protein because your cell's so big, you need all these different proteins. It's like a bunch of people trying to make copies of that same gene via transcription, and the copy machine is full. Like there's 4 people lined up trying to make copies. Like, but I need that gene because we need this protein for our organism to survive. That's why the DNA kind of limits the size of it, because it only has so much information. A lot of it, but we know how much of that DNA is junk. Do you remember? 97%. 97% is junk. Am I still in frame over here?
You or the...
Me.
I can get you right.
No, it's okay. I was just curious because I was just doing this. I didn't know if that was in there or not.
Okay.
Whatever.
Now it is.
Now it is. Now I'm out, right? Now I'm out of frame, obviously. The nucleus can only control a limited amount of active cytoplasm. So the cell can't get too big. That was my printer analogy, copier analogy. The plans are all in the DNA. You have to transcribe a protein via protein synthesis, so you have to go transcription. So you have to copy that. So when you copy that, if there's multiple people trying to copy the same section of DNA, you're going to have problems. That's why a cell can't be humongous. One of the reasons. Yes, sir?
Well, then why does the nucleus just get bigger with the cell?
Why doesn't it? Well, then you don't have other room for the organelles. Then it can't function. So it's like having too much, like a hoarder of information. So you can't actually do anything with that information because you have so much of it. Yeah. Everybody good there? Nobody's hand has died from cramping yet?
No, it's
getting there? Okay. Lots of potassium, eat a banana. You'll be fine. Okay. Alright. I still will talk about, lactic acid, anaerobic cell respiration. Good. I still will talk about excrement on this video. I'm not going to wuss out. Alright. The size of the membrane. The size of the cells obtain their nutrients and get rid of their waste through the cell membrane. I'm coming back. You don't have to move a mess for while you're writing this down. Most people think that a cell is, keep in mind that when we talk about a cell, when I'm talking about the cell membrane, I'm talking about all the blue here. This is all the cell membrane. We take a 3-dimensional object and put it in 2 dimensions on the board. People get kind of confused. Well, doesn't the cell look like an egg? Doesn't the cell kind of look like this? But imagine there's going to be a cell membrane covering it because we're taking a 3-dimensional object, putting it on 2 dimensions. We have to be able to see the anatomy of it. We need to do it like that. Hopefully, so the cell membrane, the outside, that's the gatekeeper, right? Obtains the nutrients. So this is where it gets easier, right? Oh, don't have to write that down. That's good, right? Don't freak out. As the cell increases in size, the need for the materials as well as the waste production increases. So the surface area of the cell membrane must increase in order to bring enough materials and get rid of the waste. Let me explain that into real talk. This is my excrement analogy. Super corny. Layers there, layers. Okay, so the way I look at it, guys, as the cell size increases, the amount of waste. Let's say you live like I do, live in the city of St. Louis, and you're walking a dog, and you can't just let the dog go to the bathroom wherever, you actually have to pick it up. So what would you rather do? Would you rather walk a Great Dane, or would you rather walk a teacup Chihuahua in the city? Teacup Chihuahua, right? Because, like I say Chihuahua, because the amount of waste. So as something gets larger, it's only common sense that there's going to be more waste product produced. So as the cell gets larger, it has to get stuff out of that cell membrane and it has to get stuff in. And we're going to learn that it's not very good at doing that when it's real big. And that's what the lab's for on Friday. Good? I don't know if that analogy helps, but just think, that's just intuitive sense, really. So the size of the cell membrane again. As the cell gets larger, the surface area of the cell membrane to volume of the cell gets smaller. This is important. This is kind of like the whole reason that what we're talking about today. As the cell gets larger, the surface area to volume ratio gets smaller. So if the cell is small, it has a better chance exchanging its enough materials to support its cytoplasm. Let me explain this. As the cell gets larger, the surface area to volume ratio gets smaller. Let's say I have a cell here. We're going to talk about this. And I'm imagining that the cell is a perfect cube for mathematical representation here. Let's say this cell is 1 centimeter by 1 centimeter by 1 centimeter. Right? How do I calculate the surface area of this guy? First off, how do I calculate surface area in general?
You would multiply one side and then like figure out what the area is on one side of that and do it for one side of it, right?
Right. Translation, mathematical speak. Length times width is surface area, correct? And then how many, so that'd be one side of this. Times the number of sides. 6. How'd you know that? It's a cube. Well, I just remember for me, it's not obvious to some people that there's 6 sides on a dice. That's why. Unless you're playing Dungeons and Dragons and it's like weird, like 21 or something. 30 something. Okay? We're not talking about Dungeons and Dragons. Alright, so length times width times 6. So what would my surface area be here? 6. But what are my units? Squared. Because I'm just doing boom, boom, length times width. Now, that is my surface area. You're on it, right? Very good. What's my volume? How do you calculate volume? Length times width times height. So what's my volume here?
120.
Centimeters cubed. So my surface area to volume ratio is this. 6 to 1. Also written 6 to 1. Notice that is a pretty large ratio. So this is saying that for this much cell membrane, the surface area of the cell membrane, this is how much it actually can, this is how much the volume of the inside of the cell. So keep this in mind. The cube would be all the sides. This number would be all the sides of this cube here. The 1 would be the area inside that cube. This is where it's kind of difficult. I forgot to say something also, but that's alright at the very beginning about how tiny cells are. So keep this in mind. Questions on this? Let me do this real fast. I want to expand upon this. So let's say I go back to here and I say that this is another cell is 4 by 4 by 4. Okay? Surface area is going to be, so 4 times what? 6, right? So we got 24 times 4. You guys okay with that? You guys agree with that?
Yes.
Is that a good number? Yes? Am I just making, I should start doing this incorrectly.
24 times 4.
That is not right.
That is not right. 6 times 4 is 24. 24 times 4, 16. That's right, isn't it?
96.
Oh, it's an 8. That's an 8, that's a 9. Okay, that was caught on camera, right, guys? Yeah, Mr. Smith can't do math. Sweet. He's teaching. Awesome. So, glad. So 96. We okay with that number?
Yes.
Okay. That should be surface area. Now my volume is going to be what? 4 times 4 times 4, right? Length times width times height. Which is, I know this one, 64. 16 times 4. And that's going to be... Now, somebody heard somebody yawning. Oh, math. Can't get away from it, guys. So if I reduce this, can somebody reduce this quickly? The last hour I thought
48 over 32.
Okay, 40. Can we reduce that again?
6 over 4.
Can we reduce that again?
Yeah,
3 over 2, right? All we're doing is reducing this fraction. Check this out, though. Look at this. Compare. This was 4 by 4 by 4, 3 to 2. As the cell got larger, this ratio got smaller. That's the important thing. That's the whole take-home point of this. Is that if it's too big, it cannot...
Letter?
You know what?
Oh,
Okay.
It's all on camera.
It is all on camera. Yes, sir.
What happens if the ratio gets to be 1 to 1? If the ratio is just 1? I mean, is the cell going to pop?
Interesting question. It's more complicated than that, but there is a ratio to where cells can't get too. If they get too big, they can't support the life, and that's why I forgot to mention that at the very beginning on your notes it says typical cells are within what range of a size, and it should be within 10 micrometers to 100 micrometers. All right. So that's the typical range of eukaryotic cells. Did you guys see that in your notes at the very beginning? 10 micrometers to 100 micrometers. I forgot to do that at the very beginning.
10 and 100.
10 micrometers and 100. I'll explain this in a second. And to give you, this is a typical eukaryotic cell, the size. In 1 meter is equal to 10 to the minus 6, sorry, 1 micron is equal to 10 to the minus 6 meters. Like, that doesn't make no sense to me, right? This will make sense to you. In 1 millimeter, there are 1,000 microns. So, for perspective here, because I use micrometers in the lab I used to work at, we used to cut brain tissue 70 microns thin. I would say thick, but really it's thin. So if you look here, you guys can't see it. Everybody made fun of me first hour. The space in between, from right there to there would be 1,000 microns. That's pretty crazy. So I used to cut brain tissue 70 microns thin. It's pretty thin. We could do that so we could stain stuff. So how? We had a certain machine called a vibratome, and it just moved a razor blade that went like this and cut the brain along at a very specific plane, and it just, you mess with the wheel, and it just lowered it down, I mean, very small increments. It was kind of cool. So you can actually stain the brain. Right. Right? So that's how the stain the brain. Stain the brain. There you go. Grab that. All right. We are almost.
Sorry, Mr. Smith.
It's okay. It's not like we're recording this or anything. I'm kidding. I'm kidding. I'm kidding. I'm kidding. We didn't get as far as I wanted to today, but that is all right. I almost said okay. Decided not to. So I will not. You just said it. I will not be here tomorrow, so make sure you're on your best behavior like you always are. If there's a book laying down there, I know you didn't do that, Prater. Will you please put that book up? Same thing for you guys that have books out. You can go ahead and turn it off. Mess